out of those holes. That's called hydrostatic pressure. So hydro is water. Pressure is just, if you turn on the hose, there's pressure, there's water pushing. Things are bound to come out of these holes. So that's called hydrostatic pressure. So it is passive. Again, there is no energy to move that. Same thing here. As water goes through, gravity is pulling this pressure of the water. Okay, this is a hydrostatic pressure. Okay, later on we also, I forget one or two, we're going to learn osmotic pressure, which comes from osmosis. Okay, so just remember this is hydrostatic pressure, or hydro, pushing of movement of solution causes things to come out. So that was passive transport. So things are going through. Okay, same as the oxygen and the carbon dioxide going through. That's passive diffusion. But now, when your body needs energy to move certain things across, that's called active transport. You need energy, you need a carrier, like a pump, like we just saw. It's against the concentration gradient. It's actually go from low to high. Okay, let me show it to you. Okay. For example, if we're trying to get through 20 people trying to get through a door. Okay. And 20 people are outside, they're trying to get through the door. That's very easy, if it's passive, they're just going to, slowly push through. But if you're going to go the other way, pushing into the crowd, that's active transport. Okay? From a low concentration to a high concentration where the crowd is. So we're going to look at this picture. So again, on here is the outside, we call it extracellular. Inside, on the bottom here is the intracellular. You see sodium out here. There's a lot of sodium. Okay, but inside here is only a couple of sodium. So naturally, sodium would want to come in. But because we need sodium to be outside, this goes into their special transport channel and then get pushed out. Okay, it uses energy. The energy is the ATP. It pushes out. Okay, same thing here is potassium. We're going to learn all those later when we look through muscles and nerve. It needs this type of transport for nerve impulses to go through. It creates a gradient. We'll go through it later. So again, outside potassium, there is very little potassium. Inside here, there's a lot of potassium. Normally, they would want to go out, but here is a potassium channel. It will from a low concentration to a high concentration, so it will actively transport it this way. Now, what happens if the molecule is too big and it cannot get through these channels? There's something called endocytosis. Endo is inside. Cytosis is cell. Okay, so it requires ATP when the thing is too large. How does it work? I'll show it to you. So here's the cell. There's something there. What it does is it will pinch a little hole like that. Okay, it will engulf it and bring it into the cell. Okay, again, here's something. It comes in, engulf it. Okay, and then bring it into the cell. It makes a little thing called a vesicle, which is right there, formation of a vesicle. Okay, it carries it into the cell. three types of endocytosis: pinocytosis, when there's uptakes of fluid; phagocytosis, we're going to learn this a lot when we talk about immunity; uptake of solid, when they take in a virus or something, bacteria. Receptor mediated is more only when it's activated by a receptor. Okay. Whenever there is endocytosis, there is exocytosis. Exo is getting it out. Okay. Kind of like pooping it out. Okay. Again, it requires ATP. Usually the exocytosis is when you your body made something, and for example a protein, and it will send it to other cells, whether it's nearby or far away. So it does it like that. Okay. Membrane bound sac and are released from the cells. Okay. Endocytosis is reversed, so it's the opposite of endocytosis. For example, a secretion, it makes enzyme, and then it will secrete it into the body, the rest of the, or blood. Hormones are made that way, some of them. So let's look at the, so we already looked at the cell membrane. Okay, now we're going to look at the cytoplasm. And different organelles. So the cell membrane is on the outside. The cytoplasm, okay, is this gel-like substance. Okay. It keeps the structure of the cell, give it, and the cytoskeleton helps maintain the shape. Let's see. And we're going to learn all these in the lab as well, and you're going to know them well. So, packaging, producing, and exporting, okay, endoplasmic reticulum. So we can shorten this using just say ER, okay. It's a series of membrane forming connected sac and tubule. There's a rough ER, we call it rough ER or smooth ER. Rough ER, let me see if I find a picture for it. Okay. So the ER, endoplasmic reticulum, looks like these guys. Okay. Now you see those little dots there? These little dots are called ribosomes. Okay. The smooth one does not have ribosomes on it. Here's a smooth ER. And here are some rough ER. Okay, it's like sandpaper. It only has additional ribosomes on it. Now the ribosomes are those little purple things, okay, those little dots. Ribosomes build proteins. Okay, so they're called, that's why the rough ER helps with protein synthesis. The smooth ER, because it's so smooth, just think of fat, butter, okay, very smooth, is for lipid synthesis, detoxification, and it stores calcium. So ribosomes, builders of proteins, okay, and it is formed and packaged here in the rough ER. Then, after everything is done, some require modification and direct transport. That's where the Golgi apparatus comes in. So here it is, it's done, it's finished. Then it's sent to the Golgi apparatus, and it is refined, packaged, and then distributed outside. Again, this way of doing it is exocytosis. Okay, so here it is in the general cells. Rough ER, smooth ER. Rough ER is for protein. Smooth ER is for lipid. Okay, and when it's done forming, it goes to the Golgi apparatus, where it goes through, package, repackage, and then exocytosis to the other cells or the rest of the body. Vesicles. Vesicles are those little holes that we saw that it made, made a little container, a little bubble for things to come in, or to come in is called what? Endocytosis, and for it to go out is called exocytosis. Okay. Like we saw earlier, some of them pinch off the Golgi apparatus and then get transported out. There are other ones called the lysosome. Okay, lysosome. Let me see if I can find one for you. Here's one. Here's a lysosome. Okay. So here's a vesicle. Peroxisome is it's a substance contained in a contained in a vesicle. Okay, here's a vesicle. Here's a lysosome. Oh, excuse me. The lysosome is for example, if the cell is injured, the lysosome is released and then kills the whole cell. It destroys things. Okay. Sometimes bacteria or whatever comes in, it needs to be killed. Lysosome will go in, or a peroxisome. So one of the things about lysosome is for intracellular digester, or molecule recycle and defender, for example. Like I told you, a bacteria comes in. So the mitochondria, one of the most important organelle, looks like a kidney.